Assalamualaikum and a very good day to Sardin and our friends. Today, our group, which is Group 5, are going to present about steps and method of gene cloning. So our member consists of me, Nurul Farhana, Siti Maisara, Nur Ani, Faiza Nabila, Haifa Hamza, and Dania. So I'll pass to the next person to start our presentation.
Hello everyone, I am Ani and I will be doing the introduction for our presentation about steps and methods of gene cloning. Gene cloning is the process in which a specific gene is located and copied out of all the DNA, also known as deoxyribonucleic acid, which is the core of the DNA, and is extracted from an organism. It involves the separation of specific genes or DNA fragments from a donor cell, attaching it to a small carrier molecule called vector, and then replicating this recombination vector into a host cell. In biomedical research, cloning is broadly defined to mean the duplication of any kind of biological material for scientific study, either as a piece of DNA or an individual cell. It is commonly practiced in molecular biology labs that is used by researchers to create copies of a particular gene or downstream applications such as sequencing, mutagenation, genotype, or heterologous expression of a protein. That is all for me. I will be passing to the next person.
Hello everyone. So right now let's look into the step and methods of gene cloning. So there are basically 4 stages of gene cloning process, which is isolation, insertion, introduction, and cloning. And today I'll be talking about isolation and insertion in details. So firstly, we need to isolate 2 main components that are involved in this gene cloning process, which are the DNA molecule and also a DNA plasmid. So these 2 components must be isolated before the cloning process occurs. But before that, there are 2 methods of isolating the DNA molecules. So the first method is by using a specific restriction enzyme to cut the DNA molecules at the restriction site to produce multiple fragments. So one of the many DNA fragments produced must contain the gene of interest. And to separate the gene of interest from other fragments, a gel electrophoresis process is used by arranging the DNA fragments according to their size to locate the gene of interest and separate it from others. And the second method is by producing a complementary DNA through and the second method is by producing a complementary DNA through reverse transcription process, where in this process, the mRNA strand is used as a template to produce new DNA strands. So the isolation of DNA molecule must contain the gene of interest, which it will be cut by a restriction enzyme at the restriction site. The restriction enzyme will produce either sticky or blunt ends. The restriction enzyme will produce either sticky or blunt ends on the target gene. So as we know, since the molecule has many base pairing sequences, so these sequences actually code as restriction sites. So therefore, there will be many restriction fragments to be produced. And the next is the bacterial plasmid of an E. coli cell is the cloning vector that will be used in the gene cloning process today. So the plasmid needs to be isolated. So just an additional info, the DNA plasmid carries ampicillin resistance gene and also bacterial LAC-Z gene as the genetic markers. So the ampicillin resistance gene will make the cell will make the bacterial cell to be resistant to antibiotic ampicillin, and the LAGZ gene will help in producing beta-galactosidase enzyme, which it can hydrolyze lactose or X-gal during the cloning process later. So to cut the plasmid of the E. coli cell, we must use the same restriction enzyme that we used to cut the DNA molecule earlier. So the bacterial plasmid will be cut once at the LAGZ gene because the gene is also encodes as the restriction site. So by cutting the plasmid, we'll produce an open ring plasmid with either sticky or blunt ends. So it is very important to use the same restriction enzyme for both molecules because it is going to produce the same type of cuts that will be complementary to each other. And as a result of the isolation process, one open ring of DNA plasmid and many DNA fragments with target genes are produced with sticky or blunt ends that depends on the type of restriction enzyme used. All right, so on to the next stage, which is the insertion stage. So this stage is the insertion of target gene into the cloning vector, which is our plasmid. So the open ring bacterial plasmid, it will be mixed together with the target gene fragments. So the exposed bases on the sticky or blunt ends at both plasmid and target gene, it will attract the complementary bases at other sticky ends by forming hydrogen bonds. And the DNA fragments and plasmids will come together and DNA ligase, and with the help of DNA ligase, it will join and enclose the spaces between the fragments by forming covalent bonds between the nucleotides. So this way, recombinant plasmids are produced. And at the end of the insertion stage, there are two types of plasmids that will be produced. Firstly is the non-recombinant plasmid. These non-recombinant plasmids are the plasmids that unable to take up any DNA fragment. And second type of plasmid is the recombinant plasmids that carry the target gene. And that is all from me. I'll pass to the next person.
Okay, I am Maisara and I will explain the last two stages of gene cloning, which is the introduction and cloning stages. Okay, Haifa, can you zoom in a little bit? Okay, so for the third stage, which is the introduction, is actually the introduction of a vector into a host, because now the next step we need is to introduce the plasmid, which is the non-recombinant and recombinant, into the bacterial cell, which will allow the vector containing the foreign DNA to multiply itself. So this step is actually done by inserting the plasmids that carrying the target genes into the host cells. So the most common host cells that we use are bacteria, E. coli, because it has the most suitable condition to carry out this step. So the recombinant and non-recombinant plasmids will be mixed with the E. coli in a medium that contains calcium chloride with a brief heat stroke. Okay, so this process is called S transformation. So the calcium ions in the medium helps to induce the permeability of the bacterial cell by creating holes in the cell wall. So the host cell, which are the E. coli cell, will become more permeable and able to take up the plasmids from the surrounding. So there will be three possible outcomes, whether the host cell will contain the recombinant plasmid with the target gene, without target gene, or takes up non-recombinant plasmid, or does not take up any plasmid. Okay, so for the next stage, which is the cloning stage, it is meant by the cloning of target gene by the host cell. So in this stage, we will apply the process of amplification and screening of the target genes. Okay, so all host cells will be cultured in a agar medium that contains ampicillin antibiotic and X-gal X-gall sugar. So the transformed E. coli bacteria that take up recombinant plasmid in the medium will be able to replicate and form colonies due to the resistance to the ampicillin, which is the antibiotic. So the bacteria which contain the non-recombinant plasmid will be able to produce beta-galactosidase enzyme due to the LAC-Z gene that is still intact. So the enzyme will hydrolyze the sugar, the X-gall sugar, that will produce the blue colonies. So this is because the added restriction sites do not affect the performance of the gene for the enzyme. So if the foreign DNA is inserted at the restriction site in the gene will not work. Therefore, if the bacteria that survive growing on the antibiotic are then grown in a medium containing the sugar, those colonies which lack of DNA will appear as blue colonies. So the other one, the bacteria with a recombinant plasmid will form white colonies due to the non-functional of the LAC-Z genes. This is because the surviving bacteria would clearly have the antibiotic resistance, meaning they must have successfully incorporated the recombinant plasmid into their cell. So the white bacteria colonies are then cultured on a larger scale to divide and produce new identical bacterial cells. So the recombinant plasmid will also replicate every time the cell divides. So it will produce more and multiple copies of the target gene. Okay, I'll pass to Faiza again for the diagram explanation.
All right, so now we have included a diagram on how gene cloning process is carried out so that our viewers can have better understanding on how the process works. So firstly, you can see there is an E. coli bacterium and also a cell which can be from any living cell. So from the E. coli bacteria, and from the E. coli bacterium, we will isolate the plasmid, and from the cell, we will isolate the DNA molecule. So both plasmid and molecule will be isolated and cut with the same restriction enzyme to produce sticky or blunt ends. So in this diagram, it showed the plasmid and the DNA molecule are cut in a staggered ends, which is the sticky end. So both plasmid and the target gene will be combined together, and with the help of DNA ligase, as I mentioned earlier, it will enclose the fragments together by forming covalent bonds. So Haifa, can you scroll down a little bit? All right. So after DNA ligase have joined together both plasmid and target gene and the DNA fragment, it will produce a complete plasmid that is produced in two types, which are the recombinant plasmid that contains the DNA fragments or the target gene, and also the non-recombinant plasmid that does not take up any DNA fragments. So I'll pass to Maisara for the next stage.
Okay, I'll explain I'll show how about my explanations just now on this diagram. So here, as you can see, like Faiza said just now, after the insertion stage, there are two types of plasmid produced, which are the recombinant and non-recombinant plasmid. So then in the next stage, which is like I said just now, the introduction stage, the both plasmid will be introduced into a host cell, which we are using E. coli. So after that, the next stage, the E. coli cells that contain the plasmids will be cultured in a medium that contain the sugar and the antibiotic, which are the ampicillin and X-gall sugar. So from here, as you can see on this diagram, the recombinant plasmid and the non-recombinant plasmid are inserted into the cell, and then they are transferred into a medium that contains the ampicillin and X-gal. So this type resulted the form of colonies, which are the two type of colonies, white colonies and blue colonies. E. coli with the recombinant plasmids will form the white colonies due to its antibiotic resistance, like I said in the explanation just now. So the other one is the cells that contain the non-recombinant plasmid will form blue colonies because it will break down the sugar into a blue compound. So as you can see on the diagram, there are like two colors of matters in the medium, which are blue and white. And lastly, the bacteria which form the colorless colonies, which are the white one, that contains the donor DNA, can be isolated for further cloning. So lastly, we can produce clones of the targeted genes, as it says, as the recombinant plasmid replicate to produce multiple copies of the target genes. Okay, I'll pass to the next person.
Okay, so right now, I'll be explaining about the discussion of our subtopic, which is step and method of gene cloning. Okay, as mentioned by my group members, about the restriction enzyme and DNA ligase. Now, I will briefly explain about these two important components involved in gene cloning.
Haifa,
Okay, let's start.
kita stop meeting dulu nak sebab dah nak habis. Okay, boleh?
Assalamualaikum and hello. So right now, I will be presenting about the discussion of our subtopic, which is step and method of gene cloning. As mentioned by my group member about restriction enzyme and DNA ligase, now I will briefly discuss about these two important components involved in gene cloning. Okay, let's start. Okay, as we all know, in gene cloning, there are many copies of pieces of DNA. In many cases, gene cloning does involve inserting the gene into a piece of circular DNA called a plasmid. Okay, so firstly, how can piece of DNA from different sources be joined together to make a single DNA molecules? Okay, there is a common method used, which is restriction enzyme and DNA ligase. A restriction enzyme, basically a DNA cutting enzyme that recognizes specific sites in DNA. Many restriction enzymes make target cuts at or near their recognition sites, producing ends with a single-stranded overhang. For DNA ligase, it joins two DNA molecules that have a matching ends. DNA ligase basically seals the gap between the molecules, forming a single piece of DNA. Okay. Now, I will go deeply about the restriction enzyme and how it's related in this subtopic. Okay, let's start. Restriction enzyme. Restriction enzymes, that is also known as restriction endonucleases, they are required for gene isolation and the synthesis of cloned DNA molecules. So basically, how does it relate with this subtopic is it used in the isolation process. Okay, so the majority of restriction enzymes identify 4 to 8 base pair sequences and hydrolyze a single phosphodiester bond on each strand. As mentioned earlier, during isolation process, restriction enzyme is used because isolated DNA needs to be cut at specific sequences using restriction enzymes. Okay, these enzymes are produced by bacteria as mechanism of innate immunity against intruding viral DNA and function as molecular scissors. Hence, after restriction enzymes identify a viral DNA, they start to break it apart by cutting it into segments at certain nucleotide sequences they recognize. Cutting DNA with restriction enzyme allows researchers to identify the size of unknown fragments relative to a standard ladder. These fragments of interest can then be further isolated and ligated into plasmid vector for genetic cloning. And then we go to the DNA ligase. Okay, basically DNA ligase is a type of enzyme. Okay, they also use the concept used for gene cloning is basically same as it used in the DNA replication. So as we learned before, in DNA replication, um, DNA ligase join together fragments of newly synthesized DNA to form seamless strand. So it actually works the same during gene cloning. If two pieces of DNA have matching ends, DNA ligase can join them together to make an unbroken molecules. Again, basically DNA ligase play its roles during the insertion process. By using ATP as its energy source, ligase catalyzes a reaction in which the phosphate group sticking off at 5 end of DNA strand is linked to the hydroxy group sticking off at 3 end of the other, and it will produce a sugar phosphate backbone. So basically, if you guys can see from this diagram, as I said before, the restoration enzyme work as a molecular scissors, and then the DNA ligase will glue the fragments, and it's also known as a molecular, can easily see it as a molecular glue. So I think that is all from me for the explanation about the restoration enzyme and DNA ligase. Thank you.
Okay, hello everyone, my name is Dania, and now I'm going to be discussing about the various types of vectors that are present in gene cloning. So, first off, we have plasmid vectors. So, plasmids are small extrachromosomal circular DNA molecules which are capable of replicating autonomously within the host cell. They are also termed as the workhorse cloning vector in recombinant DNA technology. Plasmids are widely used as vectors in all three domains of life. However, these are frequently used in bacteria and yeast. The most important feature of plasmids that make them one of the best vectors is their small size. The small size of the plasmid facilitates the separation of recombinant DNA from the host genomic DNA. The size of the plasmids range from a few thousand base pairs to more than 100 kilobases. The small size of the vectors does, however, affect the maximum size of the insert DNA that it can carry. Plasmids can carry insert DNA that is less than 20 kilobases as the cloning efficiency and plasmid stability decrease with the size of the vectors. The autonomous replication of plasmid is made possible by the presence of genes and sequences that can initiate plasmid replication independent of the host replication cycle. Bacterial plasmids contain ORI sequences that not only control plasmid replication, but also determine the possibility of two plasmids coexisting within the same host cell. Different plasmids have different types of selective markers, but the most common markers include antibiotic resistance and the production of the beta-galactosidase enzyme. Some of the most widely used plasmids are PBR322, PUC, and P-BlueScript vectors that use E. coli as the host. Second, we have cosmid. So, cosmid vectors are hybrid vectors that are composed of plasmid and phage lambda vectors, which are capable of incorporating up to 42 kilobases of DNA. Cosmic vectors are prepared by the insertion of the cos region of the phage vector into the plasmid vectors. Cosmid vectors are large-sized vectors with sizes ranging from 400 base pairs to 30 kilobases. These can carry DNA sequences having sizes ranging from 28 to 46 kilobases. Cosmid vectors are created in order to incorporate large-sized DNA molecules that cannot be carried by plasmids. Since these are hybrid vectors, these can replicate within the host cell like plasmids or remain packaged like a phage. Cosmid vectors that do not have many phage characteristics except the signal sequences promote phage head stuffing. The hybrid sequence of the cosmid enables the phage heads to be incorporated within all donor DNA for transfer. The use and production of cosmid vectors have increased over the years as the package system is highly efficient and selective for the recovery of larger hybrids. One of the examples of the cosmid vectors prepared and used in the practice of are cosmid PHC79, which are cos-containing derivative of the vector PBR322. Third, we have bacteriophage vector. So bacteriophage vectors are viruses that only infect bacteria and transform them efficiently while carrying large insects. Bacteriophages or phages have higher transformation efficiencies, which can increase the chances of recovering a clone containing the recombinant DNA segments. The most important feature of a phage is the packaging system, which enables the incorporation of large eukaryotic genes and their regulatory elements. The use of phages also facilitates the isolation of larger quantities of DNA that can be used for the analysis of insert. It can selectively package a chromosome about 50 kilobases in length, and the size of the phage can be adjusted by removing the central part of the genome. Then we have the human artificial chromosome. So, human artificial chromosome, also known as HAC, are extra chromosomal DNA fragments that act as a new chromosome within the human cell. The use of the human artificial chromosome has increased with the advances in genetic engineering, as it helps overcome problems commonly associated with traditional vector systems. HACs can exist as single copy episomes without integration into the host chromosomes, allowing long-term stable maintenance. Besides, there is no upper limit in the size of the DNA that it can insert into the HAC, as entire genomic units can be used to mimic the natural gene expression. Despite numerous advantages, HACs has only been used for studies related to the structure and function of human kinetochores. Limitations associated with HACs are due to technical difficulties during gene loading and ill-defined structures of the vectors. After that, now I'll be talking about the ethics discussion, so gene cloning is one of the technological advancements in medicine that has received both praise and backlash from communities all around the globe. Some speculate that if personal information of subjects are involuntarily given, their rights for their own privacy and choice to make well-informed medical decisions may be ignored. If this technology is used to treat diseases in embryos, some argue that this violates the rights of early-stage embryos. Others, however, argue that banning this technology violates the rights of those who benefit from this treatment. As well as potentially infringe the right of individuals, it also possesses ethical concerns. Although cloning members of endangered species will be beneficial, cloning an extinct animal exhibits negative responses due to its interference with nature. However, I believe that gene cloning can help millions suffering from disease and disorders. As we can see now, biomedical researchers are working on effective solutions regarding some major genetic disorders such as sickle cell and hemophilia. However, we must always be aware and cautious of the risks that it can pose. That's all for me. I will pass it on to the next person.
Okay, thank you, Dania, for your explanation. So I will explain about conclusion. So for decades, we have been using DNA ligase for many uses, and one of most important uses is in the recombinant DNA molecules, which we are discussing right now. And then the second one is we use the DNA ligase to identify mutations using the method of ligase chain reaction. So ligase chain reaction is known as a method after the polymerase chain reaction that is efficient, more sensitive and specific to detecting mutations, especially for single nucleotide polymorphisms, which is SNPS. So many of us is not familiar with the term SNPS. SNPS is known as a mutations of single base pair. For example, SNPS replace nucleotides thymine with cytosine. So the ligase chain reaction was originally developed to detect point mutation, in which when there's a single base mismatch at the junction of the two probe molecules, there is ligation cannot happen. So this means that ligation cannot happen because the mismatch in the ligation junction prevents the reaction from happening. So this made the LCR, ligase chain reaction, as an effective tool to detect mutation. Second, ligase chain reaction is also important in many next generation system. What is next generation system? Next generation system is a technology is used to detect the order of nucleotides in single genome or targeted regions of DNA or RNA. So DNA ligase it will be used to identify the nucleotide presence at the given position in a DNA sequence. So the third is the synthesis of a gene by DNA ligase. Using a ATP as an energy source, ligase will catalyze a reaction in which the phosphate group sticking at the 5' end of one DNA strand is linked to a hydroxyl group that is sticking off at the 3' end of another DNA strand. So this reaction produce an intact sugar phosphate backbone. Lastly, as a suggestion to improvise the DNA ligase, research has been made, which is to improve the DNA ligation by selective heating of DNA ligase with a radio frequency alternating magnetic field. Basically, this research is when the DNA ligation, when the DNA ligation will utilize the heat generation of the ferromagnetic particles subjected to an AC magnetic field, which is we will be using heat generated by that particles, which from the AC magnetic particles. So we carry out the ligation of the DNA fragment with cohesive end using a type of DNA ligase, which is T4 DNA ligase, immobilized in the surface of the ferromagnetic particles, which means the DNA ligase is kind of together with the ferromagnetic particles. Ferromagnetic particles is actually particles that can retain magnetism once exposed to a magnetic field. They can demagnetize and remagnetize repeatedly. So, in this research, when a radio frequency alternating magnetic field is applied, ferromagnetic particles will release heat, and the DNA ligase, which is kind of stick with the ferromagnetic particles, is heated up and activated, and this influence the annealing of the DNA ends. So as a result, in which the ligation efficiency increased. So this research concluded that as the higher the field amplitude, the higher the efficiency of the ligation. So that's all for our group. Thank you.